Network Inference by Emily Miraldi from the Cincinnati Children's Hospital.
Yes, there it is. I don't know. There, Miraldi, V2. All right. Well, thank you very much for the opportunity to give this talk. I'm going to start off before going into single-cell transcriptional regulatory network inference, giving a little background into my lab and my interest in this topic. So I'm at Cincinnati Children's Hospital, and the focus of my group is immunoengineering. And specifically, we'd like to use mathematical modeling to learn the regulatory mechanisms that drive the immune system in health and disease. And as particular applications where it would be good to modulate the behavior of the immune system, I have some examples. So we would like to tune up the response of particular immune cell populations in the context of cancer, in the context of autoimmune diseases or inflammatory diseases or organ transplant. We would like to tune down the immune response of particular immune populations. And we'd like to be able to do this while maintaining the important homeostatic functions of the immune system. So without going too much into immunology, I'd like to highlight some work in an immune cell type known as innate lymphoid cells, which are relatively recently discovered. And despite that, they have already been shown to play important roles in health and disease. And so they seem like an important cell type to focus on in terms of immunoengineering. And our goal, and this is kind of a vignette, is that we'd like to understand innate lymphoid cell ILC behavior and control in vivo. We'd like to know, in their in vivo setting, what are they doing and how are they doing that? And as a first approximation of cellular behavior, you can use RNA-seq as a high-dimensional phenotype of, what are the cells doing. And from there, we use transcriptional regulatory network inference on these measurements to try to determine what are the control mechanisms. And so, as I've been lucky to have other people describe, transcriptional regulatory networks describe the control of gene expression by transcription factors, protein transcription factors. And in our networks, a red edge indicates that the transcription factor promotes a gene's expression, and then blue indicates it represses that gene. And so our study design was based on isolating these ILC populations directly from the intestine, and then in this case doing population-level RNA-seq. But as I show you our methods, you'll see that a lot of them ended up being useful in a single-cell context as well. And so one of the challenges with transcriptional regulatory network inference is that typically there are thousands of genes that are expressed, hundreds of transcription factors that could, are expressed and could explain the gene expression profiles. Got, upwards of 10 to the 5th transcription factor gene interactions to learn, and typically you only have a limited number of samples. And so the challenge is that many transcription factors could explain a gene's expression profile. And so a typical strategy that we and others use is when you have, say, an underpowered gene expression data set, we try to integrate as much prior knowledge and relevant information as possible in order to build a more accurate model. And in the context of transcriptional regulatory network inference, it sure would be nice to have transcription factor binding data. However, in a lot of contexts, it's simply not feasible to do, say, transcription factor ChIP-seq, which is the major way that people gain transcription factor occupancy data at genome scale, given the number of cells that are required. So in vivo settings or in some of the single-cell settings, it's not feasible to do sequential ChIP-seq of hundreds of transcription factors. Also, with newly identified cell types, you might not even know which transcription factors to look at. And so I am fortunate that a lot of previous speakers have highlighted ATAC-seq as a method to measure chromatin accessibility. And essentially, if you integrate this data type with transcription factor motif databases, you can get a guess that a transcription factor might be bound to an accessible region. And this is a great tool if you have limits on sample material and you perhaps don't know a lot about the system that you're exploring. And so just from an ATAC-seq data set, you can get an initial noisy network And this you could get, for example, if you see a putative transcription factor binding event in an attack-seq peak, then you can come up with, say, linear association rules. If the peak is plus or minus 10 kb of a gene body, well, maybe the transcription factor is bound and maybe it regulates that gene. And so this is how we arrive at this picture here. And as you can see, there are a lot of errors associated with this initial noisy network. So a motif occurrence does not mean the transcription factor is bound. We've got this linear association rule. And then, importantly, the network is incomplete because we don't have transcription factor motifs for all transcription factors. And so we use the inferolator to integrate this useful prior information source with data-driven modeling to get to an improved, more accurate transcriptional regulatory network. So just at a high level, I'm going to explain some of the features of the method. And so, as I mentioned before, the core of the inferolator is to model differential gene expression as a multivariate sum of transcription factor activities. And when we're thinking about transcription factor activities, we're thinking about the amount of active protein transcription factor in the nucleus, which you can see from our experimental design was not measured. And so, we do have TF mRNA measurements, but of course those don't necessarily correlate with active protein transcription factor. And so what we do instead is we use partial prior knowledge of a transcription factor's target genes to estimate transcription factor activities. And this ends up being an important thing, and it's described in detail in these papers here. Another thing that we do is we use an adaptive lasso penalty. So in terms of incorporating prior information, first of all, the lasso incorporates the assumption that the network is sparse. So even though there are hundreds of transcription factors that are expressed, we only expect that a handful will regulate a particular gene. And then the adaptive lasso enables us to flexibly support prior information. So transcription factor gene interactions with prior support are more likely to enter the network, but if they are not supported by the gene expression model, then that prior network gets pruned. Importantly, you can also learn new transcription factor gene interactions that were not in the initial prior, which is a good thing if you're working in new cell types. And so before applying these to these ILCs, we first decided to test in Th17 cells, where there was a lot of gene expression data. We generated attack-seq data ourselves, and then took advantage of existing resources, an independent gold standard of transcription factor knockout and transcription factor chip-seq, to basically go through a lot of parts of our pipeline to determine, was this way of estimating transcription factor activity good? How much should we trust attack-seq data prior versus the gene expression model? And so this is all summarized in this paper here. We then went on and felt like we were ready to summarize the inferolator as a cylinder and apply it to these in vivo innate lymphoid cells. And so this was recently published as well, so I'll just highlight that for the different ILC subsets, we were able to come up with core transcriptional regulatory networks that had in white boxes or transcription factors that had already been associated with those particular populations. In yellow are 2 transcription factors that we experimentally validated. And so getting back to this question of immunoengineering, once you have a transcriptional regulatory network in a new cell type, you have the opportunity to try to figure out what role a transcription factor in a particular cell type might have. And so essentially what I did was for each transcription factor in the network, I tested for enrichment of the transcription factor's target genes in different phenotypes from the GWAS catalog. And what I found was that BCL6 targets were associated with inflammatory bowel disease, which is an autoimmune disease affecting the intestine. And interestingly, the ILC-specific BCL6 knockout mice that we used to validate the network had IBD phenotypes. For example, their colon length was shorter in the ILC-specific BCL6 knockout mice. So this was interesting and suggested that these networks of ILCs could be useful in thinking about human disease. However, it is quite a leap, knowing the complexity of a disease like inflammatory bowel disease, where you have a role for host genetics, multiple immune cell types, the microbiome and environmental factors. And so what we decided to do next was to infer transcriptional regulatory networks in the disease-relevant setting. And so this is teaming up with 2 researchers at Cincinnati Children's Hospital. And if it was a challenge to infer transcriptional regulatory networks in vivo in the intestine of mice, it's an even bigger challenge if you have limits of, say, 10 to the 3rd ILCs per tissue sample from an IBD patient. Also, we needed to have a method that could generate accurate transcriptional regulatory networks from single-cell RNA-seq. As you know, single-cell RNA-seq is notoriously noisy. And I think my favorite statistic when I'm trying to explain to people is that only 5 to 20% of the mRNAs in a cell go on to be captured for cDNA synthesis in current single-cell RNA-seq protocols. So, gene expression spans 3 orders of magnitude, and essentially for lowly expressed genes, this loss of, say, 80 to 95% of the transcripts is quite severe, resulting in a sparse data matrix, where, say, 90% of the entries are 0, and you don't know if that 0 is there because that gene was not detected in the cell or if it was not expressed. However, there's a lot of reason for hope. We should be able to, in theory, borrow information from both cells because we expect that there are cellular populations inside of it. Not every cell is unique. We also expect that we should be able to borrow in the genes dimension because genes are controlled, say, by transcriptional regulatory networks and co-regulated by transcription factors. And so you can see that our inference method for single-cell transcriptional regulatory network inference looks pretty similar, except we've included a step where we impute the so-called dropout genes that were not detected. And so I'd just like to briefly walk you through this pipeline. It relies on the method CIDR, which I liked because it was conservative. So you can consider the distance between 2 cells, and if you're looking at a particular gene, there are 3 things that can happen. First of all, in both cells, the gene is expressed. So you just calculate distance the normal way. And then if the gene is not expressed in either of the cells, then you don't attempt to impute. And in this context, where you have expression in one cell but not the other, you rely on an empirical distribution of the dropout rate as a function of mean gene expression to determine how to impute. And so it's a nice conservative method. And then from there, we use a radial basis function with a fixed width to generate a cell-cell similarity matrix. And this is in contrast to the method MAGIC, which uses an adaptive kernel. And essentially what this does is if there is one weird cell in your data set that doesn't have any nearest neighbors, we're not going to be imputing gene expression or knockout in there. And then the final step is then to take a weighted nearest neighbor average based on this cell-cell similarity matrix. And then the second part of our pipeline is, I think of as amplifying biological signal, and I already described it to you, and it was validated in 2 mammalian cell types. And so now, how does it perform? And so one of the things that we had trouble with were developing benchmarks. And so we took advantage of the existing Th17 resources, in particular this gold standard I described before, and combined it with 10,000 single-cell RNA-seq cell samples with Brian Bryson at MIT. And so we were able to compare our single-cell transcriptional regulatory network inference method to 2 existing methods, both of which wisely use prior information. And as you can see, the methods that I described do lead to a boost in performance. And we also compared to Pearson correlation, which came up this morning, and we outcompete that as well. what we did once we had our pipeline set up was we were able to turn off each part of the pipeline, so we could see how the method performed if we didn't reinforce with the adaptive lasso, if we didn't use transcription factor activity estimation, or if we didn't impute. And you can see that here in this context, each step does matter. We also tried to replicate our results, and so in the context of hematopoiesis, so this is an in vivo setting and a data set involving about 10 different cell types. Our gold standard is pretty small. It's only 5 transcription factor knockouts, but we did see similar results. Again, we also did experiments where we removed each of the steps, and you can see that using all of the steps in our pipeline results in the best performance in hematopoiesis as well. All right, and then the last thing that I'll mention, and this kind of echoes some results from this earlier work in, Th17 bulk cells, where we tested, what happens if you don't have access to a context-specific source of prior information like attack-seq. And so here's just an example where we're looking at transcription factor gene interactions from the TRUST database and using that as a prior. And these are generic transcription factor gene interactions that are literature curated. And you can see here it doesn't perform quite as well as the attack-seq prior, but it's not bad. And so you can still benefit from a prior-based method, even if you don't have context-specific attack-seq. All right, so conclusions and future directions. So we have this single-cell transcriptional regulatory network inference method that uses a conservative imputation method for the single-cell RNA-seq. We find that integrating prior information, just as we found in population-level transcriptional regulatory network inference using transcription factor activity estimation, and in this case, adaptive lasso to flexibly support interactions with prior support works well. Next steps, we are trying to put together something that we call the max attack pipeline. We feel like scanning accessible chromatin with transcription factor motifs, there have been so many developments in terms of the DREAM ENCODE challenge results with supervised methods that we think there are a number of places where we can get better information from attack-seq, get a better prior, and hopefully also a better network. All right, and then another thing that we're focusing on is building genetically aware transcriptional regulatory network methods, which I'd be happy to talk about. And then we're eager, now that we have benchmarked these methods, to apply them to human immunology. All right, and then with that, I would like to thank my lab, and then also thank my postdoc mentors who played seminal roles in the Th17 population level work and the ILC work, and then my new collaborators on these directions.
We have time for a couple of questions. Oh, okay, go ahead.
Hi.
Hello.
Thanks for the talk. So as the output of the activity of a regulatory network, you use expression data. But the expression data also will be affected by regulatory RNAs. So do you think it will improve model prediction?
Yeah, no, I think it would be wonderful to include regulatory RNAs. And there's some work in Rich Bonneau's group, who's there, where they've looked at the role of miRNAs as predictors of gene expression. So I think that's a new direction, and it's an important factor, so it should be accounted for in the model. Yes.
Yeah, great work. I was wondering, so your regulatory networks are very condition-specific, I assume. I mean, very
Yes,
cell-type specific. So do you also have idea of, because these cells are often stimulated, and there's a lot of kind of stuff going on upon stimulation. So do you have any data of how they change, how these networks, like, do you think they're stable when they get stimulated, or they're going to get rewired within the same cell? Do you have any data on that?
So for the population Th17 data set, we did have a lot of stimulation conditions. So naive CD4 T cells receiving TGF-beta, IL-6, and TCR stimulation, and a time course. We also had Th0, Th2, Treg, Th1. So we had a pretty big data matrix with a lot of different conditions. And
so could you predict the response from the steady states to the stimulations?
So we do, actually. We do some out-of-sample prediction, and So yes,
And
that's
works
in the
well?
paper, and it works pretty well. I think R squared of predictions. It's been a while since I've looked at it. Reach about 0.6,
Oh,
so
okay.
that's not too bad.
Oh, yeah, very cool. Thanks.
Mm-hmm.
So I have a question. It's great that you did validation, but I just want a little bit more detail about that. So was it a validation of the importance of a regulator, or was it testing the edges of the regulator, and what was the accuracy?
Yeah, so our gold standard was transcription factor gene interactions. And so for Th17, something made it into our gold standard if it was supported by both transcription factor ChIP-seq
No, I mean the experimental validation that you published in Immunity.
Oh, in Immunity. Okay, so we did a functional validation for both MAF and BCL6, where we looked at ILC-specific transcription factor knockouts, and indeed, those transcription factors were important in the cell types predicted. Only for BCL6, and I wish I had it for MAF as well, did we have RNA-seq. And so there we were able to validate based on prediction of knockout. And we had 3 cell types, ILC1s, NK, and ILC3s, where we were able to test how well we did at predicting BCL6 targets.
Great, awesome, thank you. All right, let's thank Emily once again.